Today we are going to study about mutation. Mutations. What are mutations basically? Mutations are the abnormal changes which take place at the chromosomal or gene level, and these changes may be harmful or useful for the particular organism. In most of the cases, these changes can be harmful, but in some of the cases, mutations can be useful also.
Can I go?
If we talk about the types of mutations, there are several types of mutations on the basis of whether they occur on their own or whether there are any reasons or mutation causing agents. They can be of two types. The first type is spontaneous mutations, and the second type is induced mutations. So when we talk about spontaneous mutations are the one in which the mutations occur on their own, whereas induced mutations are the one in which mutations can be induced. Now, for inducing mutations, there are certain agents which are called as mutagens. These mutagens can be of two kinds. So first, let's study what are mutagens. These mutagens are the mutation causing agents. These mutation causing agents can be of two types. They can be chemical mutagens and we can have physical mutagens. So chemical mutagens, these include the chemicals which can cause mutations. Chemical mutagens, for example, we have 5-bromo-uracil, then ethidium bromide, as you all are familiar with. This ethidium bromide is very popular. As you all know, this ethidium bromide is used for intercalating with the bases of DNA in agarose gel electrophoresis. Whenever we are preparing an agarose gel electrophoresis, whenever we want to see the bands of DNA, we use ETBr in the gel. This ETBr is added because it intercalates with the bases of DNA, and hence the bands of DNA, they appear or they fluoresce in orange color. So this is how it appears, and here you can see that this ethidium bromide is also causing mutation. Hence, it can be called as a chemical mutagen. Then there is one more chemical, you must have heard about its name. It is acridine orange. Now this also causes mutation, so this is also a chemical mutagen. Talking about the physical mutagens, we are going to study about physical mutagens. So let's see. Physical mutagens include UV rays, that is ultraviolet rays, X-rays, as well as the ionizing radiations. Ionizing radiations, I hope you all are familiar with. These radiations are basically the radiations which can cause ionization, like alpha rays, beta rays, and gamma radiations. So these are very harmful for the normal cells also. So these are the chemical mutagens and these are the physical mutagens. Even X-rays, the exposure to X-rays for a long time can also cause mutations. That's why it is said that X-ray exposure should be kept to minimum even while a person is going for X-ray diagnosis. Now, talking about another type of mutation, you can see there are some other kinds of mutations as well. Like we have two kinds of mutations, like if the mutation is taking place at the gene level, it is called as gene mutation, and if it is taking place at the chromosomal level, it is called as chromosomal mutation. Now, when we talk about gene mutation, this is also called as point mutation. Basically, this gene mutation is the change in the structure of gene. Whereas when we talk about chromosomal mutations, we need to understand that chromosomal mutations basically consist of two types. The first one is structural, and the second one is numerical. Talking about the structural mutations basically can be of several types. If there is a deletion in any part of the chromosome, it can cause the changes at the level of chromosome at the level of chromosome, and this can be called as deletion, whereas the second one is duplication, the third one is inversion, and the fourth one is translocation. So here you can see this talking about deletion. If there is a sequence like A, B, C, D, E, F, G, H, and that is present on one arm of a chromosome, and there is another arm of a chromosome like this, at which we have I, J, K, L, M. So these are basically representing the genes which are present on the chromosome. So if there is deletion, it means that one part may be deleted. That is, suppose this gene is deleted, so it is A, B, D, E, F, G, H. If there is duplication, it means that suppose it is A, B, B, C, D, E. So this is basically the duplication. You can see that. And then if it is inversion, it means suppose it is A, B, C, D, and it can be F, E. So here you can see instead of EF, it is FE. So it has been inverted. Similarly, translocation, in which one part gets translocated over to the other part. So these are basically the structural mutations. And then we move on to numerical mutations. So talking about numerical mutations, there can be two kinds of mutations. The first can be euploidy, in which the number of sets changes, but the number of chromosome inside a set remains the same. The second is aneuploidy. Now this is different. Aneuploidy means that there is an abnormal number of chromosomes inside a set. That is, the number of chromosomes inside a set changes. So here you can see euploidy can also be called in a sense as polyploidy. And talking about polyploidy, it means, for example, we talk about wheat, which is 6N, that is hexaploid. Banana, it can be 6N, that is hexaploid. If a plant is 4N, that is tetraploid. It is an example of polyploidy. So these are the cases, and these are prominent in plants. Whereas if we talk about aneuploidy, aneuploidy means the number of chromosomes inside a set has been, changed, whether it can be increased or it is decreased. So let's talk about aneuploidy, but for that I will need more space. That's why I will rub this. I hope it is clear. Okay, so talking about this, you can see this. Talking about aneuploidy, you can see this. It is basically hypoploidy and hyperploidy. So just have a look at this, students. Hyperploidy and hypoploidy. Hyper means more. Hypo means less. So hyperploidy is basically the ploidy in which the chromosome number inside a set decreases. So it can be of two kinds. First is monosomy. It is monosomy, and the other one is trisomy. Similarly, in hyperploidy, we have the increase in the number of chromosomes inside a set. So talking about human beings, we are basically diploid animals, and we have 2n number of chromosomes. So in hyperploidy, we have here, no, this was not trisomy, excuse me, this is nullisomy, and this was monosomy. Here you can see that. So nullisomy, in which it is 2n minus 2. We know that every chromosome is present in a pair in human beings. So one of the chromosome is not present in any one copy even. So even the one copy of that particular chromosome is not present. So that chromosome is completely lost from the set, and that chromosome is not having even one copy. And in monosomy, one of the chromosome, all chromosomes are present in pairs, but one of the chromosome is present in only one copy. Similarly, in hyperploidy, we have here trisomy and tetrasomy. So in tetrasomy you can see it is 2n plus 2, and in trisomy it is 2n plus 1. For example, one of the chromosome is present in 3 copies. Trisomy means that one of the chromosome is present in 3 copies, and in tetrasomy one of the chromosome is present in 4 copies. So these different kinds of aneuploidies lead to different kinds of disorders like Klinefelter syndrome. For example, it is a kind of trisomy XXY. Here the person is a male, but it is a sterile male. Similarly, in monosomy, it can be XO, that is Turner syndrome. And similarly, in a kind of trisomy can be Down syndrome, where 21st chromosome can be present in 3 copies. So all these things are there. So you should know about all these syndromes, and these syndromes have specific symptoms. For example, Klinefelter syndrome, if a person is suffering from Klinefelter syndrome, he is phenotypically a male but a sterile male, is taller in stature, but even the breast is little bit developed. If a person is having trisomy of 21st chromosome, this is called as Down syndrome, but here the person is called as a mongoloid idiot. This is a kind of mongoloid idiocy. The person is having a webbed neck, and he is mentally retarded. Similarly, in Turner syndrome, we have XO. So one of the sex chromosome is less in number. It is not present in pair. It is present only in 1 copy. So this is a kind of disorder in which the person would be a female, but she is a sterile female. She is not a normal female, and the rudimentary ovaries and rudimentary breasts are present. With this, we have come to the end of this lecture. Thank you.